The development of vaccines against infectious organisms is a remarkable accomplishment that has changed the course of human history by decreasing disease burden. Childhood diseases such as measles and chickenpox are rare today because of effective vaccines. Polio, which took a devastating toll on children and young adults 50 years ago, is close to being eradicated. Smallpox has been eradicated. The development of the smallpox vaccine is a particularly notable achievement because it preceded basic knowledge of either immunology or microbiology. So we'll focus on it. The virus that causes smallpox is a large DNA virus, variola major, that has ancient associations with human populations. Smallpox was fatal in about 30% of cases, and survivors could be identified because of the characteristic scars, pox, on their face and body from the skin lesions that typify this disease. Several important observations made during smallpox outbreaks laid the foundation for rational intervention efforts. The first was that individuals who had recovered from smallpox were resistant to subsequent infection with the smallpox virus, even if the subsequent outbreak occurred years later. The second observation was that exposure to a small amount of material from a skin pustule of an infected person provided some protection from disease to an individual who was not previously infected. What would lead medical professionals to intentionally infect a patient in order to prevent disease? The observations that recovered persons were protected from smallpox suggested that our pathogen defense system is extremely versatile at recognizing and disposing of invaders. But it takes time to develop the appropriate response during the first encounter with a pathogen. So perhaps the immune defense system could be primed by providing a small amount or a weakened version of a pathogen. Health professionals in Asia were the first to try this using an approach called variolation, which is the intentional inoculation of a small amount of dried or fresh infectious smallpox material either in the nose or skin. As humans traveled and spread smallpox, they also brought with them this potential treatment, which was well established globally by the early 1700s. Data do suggest that there was significant protection from smallpox in persons who had undergone variolation. But there were also risks. Sometimes the inoculated person developed smallpox, and sometimes other infectious agents were transmitted along with the smallpox virus. However, the process of variolation was widely used when Edward Jenner made an important third observation that laid the foundation for modern vaccinology in 1796. Cows infected with cowpox virus develop pustules on the udder, although cowpox isn't usually a serious disease in cows. Milkmaids often became infected while milking and developed pox lesions on their arms and hands. Milkmaids who had had cowpox didn't get smallpox. Thus, instead of using material from the more deadly smallpox lesions, Jenner used the less virulent cowpox material for inoculation. His test to determine if cowpox protected against smallpox challenge was successful, and despite skeptics, the change from variolation to vaccination occurred quickly. Intensive vaccination campaigns implemented in the 1960s and 70s resulted in smallpox eradication, the only human virus to be driven to extinction. With the success of the smallpox vaccine, there has been significant effort to develop vaccines against many other infectious agents. Although our contemporary vaccines are produced in a much more controlled environment, many of today's highly effective vaccines are based on similar principles to those discussed for smallpox. The two most common types of vaccines are based on intact pathogens. Live, attenuated vaccines are highly effective because they mimic an actual infection without causing the disease. This is the most natural means for the immune system to experience the pathogen, and live vaccines tend to be very efficacious. The potential problems are, of course, that some individuals may develop disease, and the agent is living, hence transmissible. Viruses attenuated to make vaccines retain the potential for rapid evolution, and so may become more virulent. Because live attenuated vaccines contain living material, they must be refrigerated, which can restrict delivery in some regions of the world. Nevertheless, live attenuated vaccines are widely used and are the basis for the highly efficacious vaccines against measles, mumps, and yellow fever. A pathogen purified for vaccine preparation can be inactivated or killed, often by heating, and then administered as a vaccine. Hepatitis A vaccine is an inactivated virus vaccine, and many vaccines against bacterial agents are based on killed organisms. Unlike live vaccines, these must stimulate the immune response without replicating. Molecular structures can be altered during the inactivation process and not recognized by immune cells. However, there is no danger of transmission or disease due to infection by killed vaccines, although there can be problems with any vaccine, either because it fails to protect or because something in the vaccine formulation elicits a negative reaction in some individuals. Although replication of the pathogen helps set the stage for the appropriate immune response, the protection conferred by vaccines is often targeted towards parts of one or several pathogen molecules. Identifying the key molecules recognized by cells of the immune system has been an important advance in vaccine development because the vaccine can be formulated to contain only the important molecules. These are called subunit vaccines because they only contain parts of the pathogen. While this is a very promising area of research, to date there are few successful subunit vaccines that have made it to market. The vaccine against hepatitis B virus is a good example. Vaccines are clearly among the advances that have improved human health and well-being globally. The concepts behind vaccination are straightforward. Jumpstart the immune system without becoming sick. Despite the many successes, there are still many diseases for which there are no vaccines. Thus, the missing details on how to prime the immune system to many pathogens provide a very active area of research.